let's get started. Happy Tuesday, everybody. We are wading through our Unit 2 material right now for our midterm exam. Just a heads up, midterm exams coming up on the Friday immediately before spring break. My goal is for you to not worry about this class, or at least the lecture portion, over break. Go have fun. Let's see here. I'm nearly done auditing all the lecture exams for number 1 that I plan on auditing. I anticipate being done very shortly with that and posting the final results. Let's see here. This week is lab exams, if you haven't gotten the memo yet. At least for my lab sections. Most of my lab sections have a modified start time. So check Canvas if you have me for a lab instructor. I posted an announcement to just my lab section saying, hey, this section starts at this time, or this section starts at the normal time, so on and so forth. Ms. Wondrich, I believe, has done the same thing for her students as well, for her lab sections. Without further ado, let's go ahead and start on our next chapter, Chapter 5 on histology. So, histology is a big deal. When we think of histology, we need to have just some dedicated time to this unit because it helps us to understand everything else. And just as a heads up, there are different schools of thought on how to teach A&P. Some A&P instructors find this to be a waste of time. And they find this chapter on histology just to be entirely skippable material, and then they cover histology on the specific organ systems as they go through the organ systems. My personal experiences have been that if we invest a little bit more time up front, everything else is easier when we cover the histology, the organ system specific histology. So we're going to dedicate a little bit of quality time today to the histology system. We have a lot of different cells in our bodies, over 50 trillion. To give you some perspective, there's an estimated, I want to say, 100 billion stars in the Milky Way galaxy. So that may or may not be meaningful to you, but we have a lot of cells in our bodies. And those cells are broken up over many different kinds of cells. We are not going to cover all 200-ish cell types in our body. Thank goodness. We'd spend forever doing that. When we think of this unit, histology, the study of tissues, we are going to focus on the tissues because tissues determine our organs, and all organs are organ systems, and organ systems us as an organism. So as we look at our tissues in histology, the tissue is typically going to have this. It's going to be a group of cells working together. In the classic definition, there's 2 or more cell types working together for a common purpose. That's pretty standard. Within these tissues, we as humans have broken the tissues up into 4 groups. Just as a heads up, there are always going to be exceptions to these categories. The exceptions make the rule. But generally speaking, all tissues can be squeezed or shoehorned into the 4 categories of epithelial, connective, nervous, and muscular. Now, one of the defining characteristics of tissues is the ratio of cells to stuff outside of the cells. When we think of the cell is the smallest living thing, the structural unit of life, functional and structural unit of life. But outside of the cell, we need something to keep them stuck together. And that thing outside of the cells that keeps cells stuck together within a tissue is called the matrix, sometimes referred to as the extracellular matrix. And within that extracellular matrix, we have proteins and we have ground substance. Now, some tissue types have lots of ground substance. Some tissue types have lots of proteins. And then some tissue types have almost no extracellular matrix at all. This is one of those defining things we need to look at to help us know what kinds of tissues we're looking at. So it all starts out in the womb. We all began as a fertilized egg, a secondary oocyte from mom, met a sperm from dad, and then we went through the primary stages of development. And during these stages of development, we had some embryonic tissues. These precursors are referred to as the germ layers. And these three germ layers are the ecto, meso, and endoderms. Ecto, as its name implies, is the external layer of tissues within the developing embryo. And these external layers give rise to things that are on the surface of our body, skin and nervous system. The endoderm is the innermost layer of tissues or cells within the developing embryo and gives rise to the stuff that is in the innermost part of our body. Think of, generally speaking, the tubes that go through us. Those tubes that go through us, the digestive tract, the respiratory tract, those tubes are going to be from the endoderm. And then we have the mesoderm for the middle-ish stuff. So when we think of the stuff in between our most superficial portion and our deepest portion, we're going to have a lot of bone, blood, muscle, cartilage in between our digestive tracts and our skin, or our respiratory tract and our skin. We spent some time on the very first week of lab on this, and it's nice where lab and lecture can overlap here. We need to focus on how to interpret tissues as we look at them. And as we're looking at tissues, it's important to understand that it's not fully representative. A lot of tissue sections have stains added that change the color of what we're looking at, usually to make it easier to look at whatever we want to look at. We use some kind of preservative. A lot of times that preservative is going to be formaldehyde, formalin, ethanol, some kind of an organic solvent. And those organic solvents, particularly the alcoholic ones, have a habit of dehydrating the tissues. If you're looking for a kind of a cool experiment, go home, take some high-proof vodka, like 180 proof vodka, and dip a wet sponge in it. And that wet sponge will oftentimes be frozen in position in that highly alcoholic solution because it dehydrates, sucks the water out of the sponge. And the same thing happens with the tissues as we're preserving them onto the microscope slides, and that dehydrating process alters the appearance slightly. And then finally we have the fixatives. The most common fixative is Permount. These fixatives are used to just glue the cover slip onto the microscope slide. And depending on the fixative that we use, sometimes they will alter the appearance, sometimes they don't. As we're looking at the sectioning of the cell, we have the three-dimensional tissues being turned into two-dimensional picture on our microscope slide. So we need to do some mental gymnastics as we're looking at these microscope slides. Let's talk about frames of reference. The nucleus is almost always going to be in the center of the cell. That's a good frame of reference. There are lots of exceptions, but generally speaking, the nucleus is that big, dark, round thing in the middle. Common exceptions include red blood cells that are anucleate, they don't have a nucleus, or maybe adipocytes, fat cells that have their nucleus kind of stuck to the side where there's this giant fat droplet in the middle of the cell. As we're looking at the slices, sometimes we're going to have a slice through the cell that completely misses the nucleus. So if we were to think of, I'll draw it on let's see how this goes. I'm going to try to draw it on the screen. So here's the cell. There's the nucleus in the middle, and just by gosh, by golly, maybe the cell gets sliced right through here. So what you would see on the microscope slide, if you were looking at this bottom section on the microscope slide, is you would see just a big circular cell membrane without a nucleus in it, because randomly the nucleus was not included in that slice of the cell. So we have to think about how the three-dimensional orientation of the cells and how the cells are sliced are going to influence the outcome of our two-dimensional picture. This is a figure, it's a classic figure. I've been seeing this figure for about 15 years. It shows the egg and the macaroni noodles being sliced in different ways. In lab, you had a banana and some spaghetti jello we had you slice up the first week, just so you can practice visualizing different slices, giving you different images of the exact same thing. And as we're slicing things, we need to have a common language. So as we're looking at microscope slides or tissue sections, we'll typically hear longitudinal. So if you're looking at a microscope slide and you see an L.S at the end, that longitudinal section means that the tissue was cut along this lengthwise axis, or longitudinal or long axis. If we have a C.S or an X.S, that stands for cross-sectional slicing. A cross-sectional slice is a transverse section through the long tubular tissue or the organ. And then finally, as we're looking at our structures, if it's some kind of a slice in between longitudinal and cross-sectional, we refer to that as the oblique slice. So we can think of an oblique as kind of a generic miscellaneous category. All the in-betweeny-weenies get categorized as oblique slices. You should make a margin note on your slides. This terminology applies typically towards tubular organs or organs or structures that can be long and skinny. For instance, you're never going to see cross-sectional or oblique slicing in terminology on a smear on your microscope slide. When we make a smear, we just take the tissues and kind of rub them around and smear them on the slide. So we no longer have any 3D orientation associated with that smear. Or if we think of adipose cells or adipose, we really don't have like a tubular structure associated with body fat. It's just there. So depending on the tissue type, we may have this longitudinal, cross-sectional, oblique terminology used. And if we don't, it's typically going to be a tissue type that doesn't come in a hollow or long-shaped structure. So as we look at the smear, that smear is going to be tissues that are rubbed across the slide. Smears are really useful in that they typically will give us one layer of cells. And it's nice to have only one layer of cells because then you can really focus on the one layer of cells and not have cells above or below obscuring your focus or making the image fuzzy as you're looking at it. I'm thinking of the areolar tissue slides that we have in lab right now. I don't know about you, but I personally struggled with finding collagen fibers on our areolar slides in lab because we have really thick slices. I mean, good gravy, they're like 100 microns thick. And for a tissue slice, that's really thick. We also could have a spread. When we think of a spread, that spread is typically going to be like the smear, but it's a less aggressive smear. So with a smear, there's going to be kind of a back and forth as you make the microscope slide. With the spread, it's more like making a sandwich and smashing the sandwich. So you have the microscope slide on the bottom, cover slip on top, and the tissue getting smashed in between when you make those spreads. It spreads the tissue out as you squeeze it. So let's focus on epithelial tissues, our first of the four categories. The first two categories we're going to talk about, epithelial and connective, are kind of beastie. There's a lot of tissues in these categories. Nervous and muscular go relatively quickly. We're going to focus on the complicated stuff first. So when we think of epithelial tissues, one thing that we really need to emphasize here is that they have very closely adhering cells. Here's another way to completely reword that. There is almost no extracellular matrix, almost no component outside of the cell. So when we're thinking of tissue types, these epithelial tissues have almost no extracellular matrix. The cell membrane of one cell is butted up against the cell membrane of the next cell. And then when we look at these epithelial tissues, they're typically going to be the coverings or the membranes, and any glandular secretion in our body is going to be also categorized as an epithelial tissue type. So the glands that make the secretion, not the secretion itself. It's also worth emphasizing that they are avascular. They are without blood vessels. So for those of you who really like looking at root words, the prefix A means without. What are they without? They are without the vasculature, or the tubes associated with the blood vessels. So if there's no blood vessels in epithelial tissues, you've got to be wondering to yourself, how do they get oxygen? We've talked about cellular respiration. We need oxygen to go to the mitochondria. We need to deliver glucose. We need to remove carbon dioxide. How do we have nutrient and waste exchange? Well, the short answer is through diffusion. These epithelial tissues are usually right next to a connective tissue that has tons of blood vessels in it. And there are so many blood vessels right next door in the connective tissue that the nutrients diffuse over to some of the layers of the epithelial tissues. Now, if we're looking at a single layer, no problem. The cells can get a lot of nutrients. If we have multiple layers, it's usually the layer or two that's right next to the connective tissue that has living cells. And then the layers of cells that are far away from the connective tissue are dead. Think of your skin. You can scratch your skin, and it doesn't hurt because the connective or the epithelial tissues on the surface are totally dead. So what do we use epithelial tissues for? We use them for protection, particularly if we have a multiple layer membrane or stratified epithelial tissue that's going to be designed to resist abrasion and also to serve to segment different parts of the body from each other. So we can think of these epithelial tissues as containing infections. When we think of our epithelial tissues, they can produce and release chemical secretions. This is the glandular functions. They can excrete waste products. You may not realize this, but some of your protein waste, as you digest proteins, are excreted onto the surface of your skin as uric acid. And it's one of the reasons why body sweat or sweaty gym socks can start to sink after a while from that waste that you sweat out. We can absorb stuff through epithelial tissues, in particular, right in the margins, with simple layers. If there's one layer of cells, it's probably going to be for some kind of absorption, whereas if it's stratified, if we have multiple layers, it's typically going to be for protection. We can filter stuff, and we can also use epithelial tissues to sense things. We can use them to conduct information. A classic example of this, and this happens in Wisconsin all the time, is you're walking around in the woods, and you feel something tickling your leg hairs, and you look down, and you see a tick crawling up the side of your leg. That's an example of the epithelial tissues of your skin giving you some sensory information, transmitting that mechanical information to your nervous system. Now, as we think of our epithelial tissues, these tissue types are typically going to be on the front line, the first line of defense in our bodies. And because they're on the first line of defense, there's typically going to be a high mortality rate or a high rate of attrition to these cell types. If you want to see how easy it is for you to lose epithelial cells, wait till it's really sunny outside and you can see a beam of sunlight going in through a window in your apartment or house, and then scratch your arm in front of that beam of sunlight, and you can see the cloud of dead skin cells rising up. You just sacrificed a couple thousand cells for that demonstration. We go through billions of epithelial cells a day. Kind of makes you think about all the dust that builds up in your house. So because we go through so many epithelial cells every day, there's going to be a high rate of mitosis. We're going to be actively reproducing and dividing these cells. Now, to connect these epithelial cells to the underlying tissue type, usually a connective tissue, we are going to need a little bit of extracellular matrix. And this particular band of extracellular matrix that connects the epithelial tissue to the other tissue type is referred to as the basement membrane. You can think of the basement membrane as rubber cement. And if you don't remember what rubber cement is, it's that glue that smells really good and it's sticky and stretchy all at the same time. So we have kind of a sticky, stretchy membrane of extracellular proteins that anchors our epithelial tissues to the other tissues that it connects to. Some common things include collagen, and you should circle collagen. There is a ton of collagen in this basement membrane. Other proteins include elastin, which makes up the elastic fibers, some glycoproteins, but collagen is the big key player. We also have a basal surface and an apical surface. And when we think of the basal surface, I want you to think of the deepest layer of cells within the epithelium. When we think of the apical surface, I want you to think of the most superficial layer of cells within a stratified epithelial tissue. Now, this is kind of a classic trick question. Sometimes A&P teachers will show you a simple cuboidal epithelium, and they'll ask what layer is the apical surface and what layer of cells is the basal surface. In those situations, if there's only one layer of cells, that one layer is both the basement and the apical surface, or the basal and the apical layer of cells at the same time. It's on top and on the bottom. Generally speaking, though, this basal and apical terminology isn't going to be used for simple epithelial tissues that typically will be used for stratified epithelial tissues. So when we think of simple, just to review, it's going to have one layer of cells. If it's stratified, it has more than one layer of cells. And of course, we have some in-betweeny categories like transitional or pseudostratified. We'll talk about those in more detail. So to name our epithelial tissues, we just need to know the system. We need to know the number of layers and the shape of the cell. And then you can mix and match. Kind of like those games you would play on the school bus where you would just mix and match nouns and verbs and come up with funny sentences. I forget what that's called. So, here are the examples. We can have, for the shapes, we can have one layer, which is simple. We can have multiple layers, which is stratified. Or the in-betweeny one, pseudo-stratified, looks like there's multiple layers, but there's really only one layer. And the key characteristic of pseudostratified is usually that the apical side of a columnar cell and the basal side of a columnar cell are going to have varying diameters, or they'll be varying widths to the top and the bottom of the cell. And because the cell isn't a uniform thickness from top to bottom, it can look like there's multiple layers. You can see in your two-dimensional slice little guys like that one right there, that little triangular cell I just circled in green, that kind of get wedged into that make it look like it's stratified even though it's not. And then for the cell shapes, we have squamous, or flattened. A side profile looks to me like, I don't know, a pancake. Top down typically is round. Cuboidal officially is supposed to look like a cube, but you've probably noticed in lab, cuboidal cells usually are more circular in appearance or have very aggressively rounded edges to the cube. And then we also have the columnar, the cells that are much longer than they are wide. Now, a key distinction between the columnar and the squamous cell types, because they both are longer than they are wide, or depending on your orientation, wider than they are long, is to focus on the amount of cytoplasm in the cell. And this is a key characteristic to focus on. When we look at these squamous cells, there's almost no cytoplasm. The nucleus makes up the vast majority of the inside of the cell. When we look at the columnar cells, there's a lot of cytoplasm all around the nucleus. So if you struggle with getting squamous and columnar mixed up with each other, focus on the amount of cytoplasm relative to the volume of the nucleus. So we can take those layering and shapes and mix and match. We can have simple squamous, which is going to be one layer of flat cells. Simple cuboidal, which is one layer of roughly spherical or cuboidal shaped cells. And we can have simple columnar, one layer of tall, narrow cells. And then there's an in-betweeny category that's really hard to spell, the pseudostratified columnar epithelium. Oh, that's a tough one to spell. And then when we think of that one, it, again, looks like it's stratified, but it's not actually stratified. We also could have, within our epithelial cells, goblet cells. In particular, goblet cells are going to oftentimes be interspersed with simple columnar or pseudostratified columnar tissue types. What do these goblet cells do? They make a carbohydrate called mucin. And then when mucin is excreted, it mixes with water and turns into mucus. So you could think of goblet cells ultimately as mucus-making cells. And these goblet cells officially are shaped like a goblet or a wine glass, although for me in particular, when I look at a microscope slide, I just look for a big, round, clear space within the tissue layer. I don't really see the wine glass. So Here is a simple squamous epithelium. On the left-hand side of your screen, you can see multiple tissue types mixed together. On the right-hand side of your screen, we have just the simple squamous epithelium being highlighted. So this layer that's highlighted is our simple squamous epithelium, and then everything else underneath that simple squamous epithelium that I'm scribbling green right now, that's just a big mess of smooth muscle fibers or smooth muscle tissues with different orientations. So one of the tough things as a student when you're looking at microscope slides is figuring out where on earth am I supposed to be looking on this microscope slide. For instance, in lab, you have a pubic symphysis slide. And last week during lab, we asked you to look at the fibrocartilage on that slide, which was right in the middle. But if you didn't know what you were looking for, there was a whole bunch of other tissue types on that slide. So one of the key things you need to focus on is to think, okay, if it's a simple squamous epithelium, it's going to be a membrane or a covering. So I should focus on the edge of the tissue section. It wouldn't be buried in the middle of a tissue section. And that's just one of those things that becomes easier as you learn structure and function and connect them with each other. Here's another one, simple cuboidal epithelium. Simple cuboidal epithelium is going to be one layer of roughly cuboidal or spherical shaped cells. We have a lot of simple cuboidal epithelium within our kidneys, and we use that to modify or filtrate as we make urine. As we think of a simple cuboidal epithelium, we can use it for both secretion and absorption, and they're all over in our bodies. Anywhere we have secretion or absorption, which is in most organs of our body, we're probably going to have a couple simple cuboidal epithelial layers. We also can have a simple columnar epithelium. When we think of simple columnar epithelium, I want you to focus on the fact that we have one layer of elongated cells. So here's the actual photomicrograph on the left, and you can see how there's these regions of white space. And these regions of white space are the goblet cells that produce the mucin, which gets mixed with water to make mucus. So when we think of the simple columnar epithelium, these cells are typically going to be in places where we need lots of absorption, lots of secretion, and we need to have lots of processing of the material that we're absorbing in secretion. Simple squamous epithelium is great for absorption, but because there's so little cytoplasm, we don't have enough organelles to really process the secretions. We just have the material go from one side of the membrane to the other side of the membrane. When we work with a simple cuboidal or simple columnar epithelium, there's a lot of cytoplasm with a lot of organelles, and we're going to process those materials as the materials are moving through the cell to get to the other side of the membrane. So where do we have simple columnar epithelium? Primarily, I want you to focus on the digestive tract. Most slides for simple columnar epithelium are going to involve the stomach, the small intestine, and the large intestine. We also have that pseudostratified columnar epithelium. I'm going to back up a slide here because simple columnar and pseudostratified columnar, these two cell types are commonly mixed up by students. So let's focus on key differences. As we look at simple columnar, the cell width on top and the cell width on bottom. So the apical side and the basal side of the cell are approximately the same width. When we look at the pseudostratified columnar, that apical side and the basal side of the cell are very different widths. The cells often have a triangular, elongated triangle appearance to them. So that's a key characteristic to focus on to help you differentiate simple columnar from pseudostratified columnar. This particular pseudostratified columnar epithelium also is loaded with goblet cells. So it's going to have a lot of mucus-making cells mixed in with it. Most pseudostratified columnar epithelium that we're going to focus on is going to come from the respiratory tract. Typically, it's going to be trachea cross-section slides or bronchi cross-sectional slides. And when we think of our respiratory tract, we produce a lot of mucus to help get dust out of the air so the dust doesn't get trapped in our lungs. And we make that mucus with the goblet cells, and then we have a bunch of cilia that get rid of the mucus. So you probably noticed that usually in the morning when most people wake up, they have to cough a little bit, and they cough up some mucus that has been building up in their lungs, and then they swallow it. So when we quote-unquote clean our air filters, we are actually swallowing dirty mucus from our lungs and sending it to our stomach to be digested. So let's focus on stratified epithelium. When we think stratified, think multiple layers of cells, 2 or more. Occasionally, we'll have over 20 layers of cells, but that's usually the exception. The one exception I can think of right now is thick skin within the epidermis. That stratum corneum has more than 20 layers of cells to it. But generally speaking, 2 to 20 layers is going to encompass the vast majority of stratified epithelial cell types or tissue types. These cells are going to rest directly against each other, which again is another way of saying that there's almost no extracellular matrix. In terms of these stratified epithelia, we can name them for the shape of the cell: squamous, cuboidal, columnar, and yeah, stratified columnar is very rare. Difficult to find that cell tissue type. And then that transitional epithelium. So, when we think of stratified squamous, that's multiple layers of flattened cells. It's very, very common because it's designed specifically to resist abrasion. Anywhere in our body where we're going to have stuff rubbing, we're going to have a stratified squamous epithelium. So on the surface of our bodies with our epidermis, we have a stratified squamous epithelium, and the cells are filled with keratin. In our mouths and our esophagus, and through large portions of our insides or our digestive tract, we're going to have stratified squamous epithelium that's non-keratinized. So if you think of chewing food and popping like tortilla chips in your mouth, you're going to have a lot of friction in your mouth as you're chewing on those tortilla chips or those honey roasted peanuts or whatever your favorite snack food is. And we need to be able to resist that abrasion in our mouth as we're chewing it. The deepest layers, known as the basal layer, is going to be exposed to the nutrients of the underlying connective tissue and have lots of cell division. So we'll have living cells at the basal layer. And then those apical layers are going to be the dead layers. And because the cells are rapidly reproducing in the basal layer, they're going to be big and fat and not really squamous shaped. But as the cells go from that basal layer and they work their way up to the apical layer, they start to lose their nuclei. And as they lose their nuclei and other organelles, they become flattened and assume that classic squamous characteristic. The two key kinds of stratified squamous are keratinized and non-keratinized. Keratinized are typically going to be on the surface and are in situations where we have high friction or we need waterproofing. Non-keratinized are going to be in our bodies, and they'll be in medium friction environments, and typically will be in areas where we don't need to worry about waterproofing. Some classic examples of non-keratinized stratified squamous epithelium include the oral cavity, the pharynx, the esophagus, or the vaginal canal. Areas where there still will be frictional forces, but not a lot, and where we don't have to worry about waterproofing. So as we look at the keratinized layers, this is a beautiful layer of some thick skin. So as we're looking at this thick skin, we have some ridges. Everything that I just circled in green is stratified squamous epithelium. And as we look at that basal layer, The cells that are right next to the underlying connective tissues are going to have lots of active mitosis. So they're going to be actively reproducing, and then as they move towards the surface, they become flatter and flatter and flatter. For me specifically, in this particular slide, when we look at the skin, I really like the stratum granulosum of the epidermis because you can still see the outline of the cell, and you can see a flattened shape. By the time we get to the stratum corneum, the cells have been so flattened that, from my perspective, when I look at these slides, it's really hard for me to distinguish one layer of cells from the next layer of cells because they've been pressed together so tightly. So as we look at our non-keratinized stratified squamous epithelium, a key difference is the visibility of the cell membrane or plasma membrane. So as we look at these tissue types, it's really easy to see the outline of the cell. So that's crystal clear as you look at the shape of that cell, whereas with a keratinized cell, there's so much keratin filling up the cells that it obscures the plasma membrane. Next we have stratified cuboidal epithelium. Multiple layers of roughly spherical cells. In lab for our stratified cuboidal epithelium, we were focusing on the mammary glands within breast tissue. Depending on where you look in the within the skin, sometimes the Endocrine sweat glands are going to be a simple cuboidal epithelium or a stratified cuboidal epithelium, depending on where the slice is in the gland. For me in particular, I like to zero in on the simple cuboidal epithelium portions of the Endocrine gland, not the stratified cuboidal epithelium portions. I like this example of an Endocrine sweat gland that we have up on the screen because it helps to illustrate the fact that sometimes you can have both within an organ. It can be simple, it can be stratified. We don't have to be very rigidly set to one or the other. Sometimes within the human body, there's going to be a spectrum of characteristics as we look at the tissues and the organs those tissues make up. And next we have transitional epithelium. This transitional epithelium can be tough to differentiate from the underlying connective tissue. I like to have my students focus on the nuclei of the cells. So as we're looking at a transitional epithelium, the nuclei are big and round within those cells. And we can see above this green line that I'm tracing, there's a layer of big, round nuclei. On the inferior margin, the nuclei are smaller and more oblique or oblong or ovoid. Pick your favorite adjective. And then above the green line, the nuclei are larger and rounder. So as we're looking at this, we could have a basal layer represented by the red line. So a layer at the bottom, or the deep layer, and then let's go with blue. Particularly with transitional epithelium that is relaxed. So it's not stretched out, but it's relaxed. When we relax the transitional epithelium, oftentimes you're going to see a scalloped or bumped appearance to the edge of the cells on the apical layer of cells within that transitional epithelium. Transitional epithelium is typically or classically located in organs that are stretchy. So the ureters, the tube that carries urine from our kidneys to the urinary bladder, and then the urinary bladder itself. These are tissue types or organs that are designed to stretch out. And the transitional epithelium is ideally suited to be stretchy. Its structure makes it ideally suited for the function of being stretchy. When we stretch out a transitional epithelium, oftentimes it'll be distended on the microscope slide. That's the term they'll use. Instead of seeing that scalloped appearance on the apical layer of cells, the apical layer of cells is completely smooth and pulled tight. So let's pause for a moment. Let's have a review question. This is a click on target. We have some transitional epithelium up on the screen. I want you to click on the apical layer of our transitional epithelium. And if you need to, you can totally talk to the person next to you. We have about 30 seconds left. Make sure you get your answers submitted at the 15-second mark, so there's time for it to be processed. So we're at less than 15 seconds. Make sure you have answer submitted. T-minus 10. All right, let's see how we did. I think these click-on-target pictures are super cool. Question, comment? What? That sounds like a device-specific issue if you're not getting a picture. So let's talk after class. So hey, cool. Most of us clicked on the surface, and it looks like most of us had a pretty centered point of view and that we clicked on right on the middle. Any layer across, let's back up a couple pictures here. Wait for it. Any layer across this surface is going to be the apical layer. So if you clicked along there, you were golden. Then I'm looking at the results. Let's see our responses. I just screwed that up. Sorry about that. Let's just move on. So if you see, when you look at the grade book as you're leaving class, if you see for this first review question that you didn't submit an answer, it's because I backed up and went forward again. So we all get full credit on this review question. So I'm just going to wave my magic wand right there. My bad. As we're looking at cell junctions that hold these cell types together, these cell junctions sometimes are going to be from cell membrane to cell membrane. Sometimes they're going to be within an extracellular matrix, and they're going to be within protein fibers embedded within a lot of ground substance of that extracellular matrix. So the cell either is going to be anchored to other cells or to the matrix. There are very few exceptions to this. Really, in my mind, the only exceptions I can think of right now are blood, semen, and lymphatic fluid. Basically liquidy tissue types. Other than that, all tissue types have some rigid structure, three-dimensional structure to them. So let's look at these interactions with each other. These interactions are important because they help to give orientation to the tissue and they allow for communication between cells. We can have tight junctions. And really, when you think of a tight junction, I just want you to think waterproof. When you hear tight junction, I want the first word that pops into your mind to be waterproof. These are the waterproofing junctions. They make it so that water cannot squeeze in between two cells that are right next to each other. Some people like to describe a tight junction as a zipper. But I see water leak through zippers all the time, so I don't like that analogy. So when we think of these tight junctions, they are going to be very small and very dense, and they are going to seal off the extracellular space. So where do we have lots of tight junctions? We have lots of tight junctions in parts of our body where we don't want liquids to move across a membrane. For example, in our stomach, we don't want digestive acids to go from in our stomach to into our abdominal pelvic cavity. That's a big, painful no-no. So we have lots of tight junctions in our stomach to help keep the digestive juices in the stomach. Or if we think of major arteries, big arteries and the big veins, we want liquid to stay in those big tubes. So we have lots of tight junctions in those major blood vessels. Another junction is a desmosome. And a desmosome is not waterproof like the tight junction. When we think of desmosomes, I want you to think of physically anchoring tissue types together. We here in the Midwest, thankfully, don't have to suffer or worry about earthquakes, and our building code reflects that. But in parts of the world that have lots of earthquakes, building code requires that they have mildly flexible connections holding buildings together, so that the parts are connected, but they can flex with the earthquake. And in the same way, we have these desmosomes that anchor the cell cell membranes together, but also allow for flexion. And if we can bend a little bit, we're much less likely to break. So these desmosomes are ideally suited to resist mechanical stresses. Now, as we're looking up at the figure on the screen, that's a full desmosome. And this is going to allow for a flexible connection that directly connects one cell to another cell. There's a subpoint on this screen, the hemidesmosome. That hemidesmosome is going to be half of a desmosome, and instead of anchoring it to another cell, it's going to anchor the cell to a basement membrane, which is that portion of the extracellular matrix that has lots and lots of collagen fibers within it. We also have gap junctions within our cells. These gap junctions are, for lack of a better term, holes. They're perforations. They make our cells like a wiffle ball, for those of you who played t-ball when you were 5 or 6 years old. So these gap junctions are just perforations in the cell membrane. They are going to be integral proteins within the plasma membrane that allow for there to be a hole in the center. And with that hole in the center, water can flow in and out. Ions can flow in and out. It's kind of like leaving the front door to your house wide open. And if you leave the front door to your house wide open, things can go in and out of your house at leisure. In the same way, with our gap junctions in our cells, if we leave those open, molecular signals can go in and out of the cell at their leisure just based on simple diffusion and concentration gradients. When we think of gap junctions, we have a lot of gap junctions in our cardiac muscle cells. We'll talk about those more later on in this presentation. That's the classic part of the body that most A&P teachers talk about, gap junctions. But other than cardiomyocytes, cardiac muscle tissue, there are gap junctions in the smooth muscle tissue, lots of our embryonic tissues, and then we think of cells in our body that need to have nutrient diffusion, but we can't have blood vessels. We'll have a lot of gap junctions there as well. In particular, the lens and cornea of our eyeballs are made of living cells. We need to have lots of nutrient exchange to keep our eyeballs functioning, the lens and cornea. But we can't have blood vessels in our lens and cornea because a blood vessel would physically block light from getting to the back of our eyeball. So we split the difference, we compromise, and we have gap junctions so that nutrients can diffuse from the cells on the edge to the cells in the middle. So here are the cell types we talked about. So that you can see a picture, the hemidesmosome is down here on the bottom of the screen, anchoring the cell to the basement membrane. Let's talk about glandular secretions. This is the part of the epithelial tissue type that gets neglected. There's not a lot of emphasis given in most A&P textbooks to this particular portion of epithelial tissues. As we look at glandular secretions, sometimes they are going to make secretions that stay in the body. Sometimes they're going to make secretions that are going to remain in the body. And when we look at these substances that are being emitted by the cells, we will say secretion and excretion. And in this context, excretion is the removal of bad molecules or waste products. Secretion is going to be the removal of something good from our cells that we are going to use somewhere else in our bodies. Depending on our current level of homeostasis and fitness, sometimes these terms will flip-flop. For example, if we're suffering from metabolic alkalosis, we are going to excrete bicarbonate ions into our urine. And in that situation, bicarbonate is a waste product. But if we're suffering from metabolic acidosis, we are going to excrete hydrogen ions into our urine. And instead of bicarbonate being a waste product, bicarbonate is something useful that we want to keep in our bloodstream. So this terminology of secretion versus excretion is a fairly fluid terminology that depends on your specific physiological condition at a specific point in time. The key thing I want to emphasize here is secretions are for good things, excretions are to get rid of bad things. Now, if we make an excretion, a secretion, if we make a secretion and that secretion goes into the bloodstream, that is going to be an endocrine secretion. So one way to think of endocrine is that it goes into the blood. Another way to think of endocrine is that it stays in the body. Endo meaning in. The most common endocrine secretion is going to be, it's going to be a hormone. And when we think of a hormone, I just want you to think of a chemical messenger that carries information from one cell to another cell, and it uses the blood vessels as the method of transmission. So if it's a molecule that carries information through the blood vessels from one cell to another cell, it's a hormone. And when we think of these hormones, we have we have hydrophobic and we can have hydrophilic hormones. We'll talk more about those in Unit 3 when we talk about the endocrine system. We have a whole chapter dedicated to endocrine glands. Common endocrine glands include the liver, gonads. When we say gonads, we're referring to sex glands. So the female sex gland is going to be the ovary, the male sex gland is going to be the testes, and then the pancreas as well. But there are many others we'll talk about. When we think of an exocrine secretion, exo means out, so that prefix exo means out. Exocrine secretions are going to go onto the surface of an epithelium. So instead of being secreted into the bloodstream, they're going to be secreted onto the apical layer of an epithelium. And I'm being kind of pedantic when I say that. I used to say secreted to the outside of the body. And for most situations, that works really well. So think of sweat glands, or sudoriferous glands, sebaceous glands, all those secretions that are on the surface. Students typically don't struggle with those. The ones that confuse students are the secretions into the digestive tract. Those secretions that go into our digestive tract are going to leave the body 12 to 24 hours later, so they are technically exocrine secretions. So when I think of these exocrine secretions, I'd like to think of a secretion onto the apical surface of an epithelium, because we have epithelium lining our digestive tract. Now, as we look at our glands, we have multiple ways of categorizing our epithelial glands. They can be unicellular, as that name implies. They are a one-cell gland, unicellular. Unicellular glands, or one-cell glands, can be either endocrine or exocrine in their function, depending on where they're located in our bodies. A classic exocrine unicellular gland is the goblet cell. A classic unicellular endocrine gland is going to be the beta cells within the pancreatic islets of our pancreas. And as we're looking at our exocrine glands, the glands that make secretions which ultimately leave our bodies, there's going to be some common terminology for these glands. We have a capsule, which is going to be the outer protective layer of the gland. We're also going to have the stroma, which is going to hold the gland together. And then within the stroma, surrounded by the capsule, we have the parenchyma. And these are generic terms. Capsule, stroma, and parenchyma are three generic terms that are used to describe many different glandular structures within our bodies. So when we think capsule, think of the protective layer on the outside. When we think of stroma, think of the connective fibers on the inside. And when we think of parenchyma, I want you to think of where the action happens, what actually makes the secretion of that particular gland. So as we're looking at these glands, we can have different categories based on their shapes, in addition to the number of cells. So these glands that we're looking at right now, these are all unicellular glands, multicellular glands, meaning there is more than one cell involved. If it is tubular, it has a long, narrow secretory portion. A cinar means that we're going to have a bulge of secretory cells, sometimes referred to as a sac. For example, this bulge right here is a sinus or alveolus. And then over here, this elongated tube is going to be a tubular secretion. Or we can mix and match and have tubular acinar. So to connect this to lab objectives, the eccrine sweat glands that you've been looking at under microscopes, those are going to be tubular, simple coiled tubes. And then when we think of the sebaceous glands within the integumentary system that are right next to the hair follicles, those sebaceous glands are going to be a compound tubuloacinar gland based on the structure of that gland type. We also can categorize our glandular secretions based on what they excrete. If it's a very watery secretion, it's going to be called serous. So serous glands are going to make secretions that have typically a very high flow rate, a very low viscosity. Classic examples include tears, saliva, and milk. When we think of mucus glands, those are going to be overwhelmingly goblet cells, making the carbohydrate mucin, and that mucin plus water gives us mucus. In case you're interested, you can buy purified mucin online. And if you take a kilogram of purified mucin and dump it in a kiddie pool, it's really fun. And then we can have the mix and match. For these mix and match glands, we're going to have, it depends on the situation. If we're very hydrated, they'll be more on the serous end of the spectrum and have more of a watery secretion. And if we're dehydrated, the secretions tend to be more mucus or high viscosity and consistency. We also can categorize our glands based on how they make their secretions. Good gravy. There's a lot of ways to categorize glands. Shape, secretion type, secretion method, number of cells. So we're going to focus on secretion method now. If we have an apocrine secretion, we are going to have a transport vesicle bud off of the cell. So apocrine secretions are going to have budding from the cell membrane. Now, if we look at the bud, or that transport vesicle leaving the cell, we are going to have whatever our favorite chemical is concentrated within that vesicle that leaves the cell. And then our favorite chemical is going to be surrounded by a bunch of phospholipids from the cell membrane. So when we think of apocrine secretions, generally speaking, apocrine secretions have a medium-ish, and I'm being very, wishy-washy here, a medium level of fat associated with them because of all the lipids that are being lost from the cell membrane to make these secretions. It's worth emphasizing when the apocrine sweat glands were first discovered and studied within the integumentary system, it was thought that they secreted via this method. It was after the fact, after they were initially discovered and well documented, that we realized they actually use a different method of secretion. But that terminology is so embedded in the literature, we still refer to apocrine sweat glands as the apocrine sweat gland, even though they use a merocrine method of secretion. So, what's merocrine? Apocrine is budding. Merocrine is exocytosis, or in a word, squirting. So instead of having the cell membrane bud off and have a transport vesicle physically leave the cell, we're going to have the cell squirt via exocytosis our favorite chemical out of the cell. That's going to be our merocrine secretion. So when you think apocrine, I want you to think budding. When you think merocrine, I want you to think squirting. And then we have holocrine. When we think of holocrine, I want you to think of self-destruction. For a holocrine secretion, the cells are going to completely disintegrate themselves. And the entire cell and all of the macromolecules of that cell are eventually going to be released from that gland. Holocrine secretions are going to contain the highest lipid content because all the phospholipids of the cell membrane, the nuclear envelope, the Golgi apparatus, all of those membrane-bound organelles are going to be released with this holocrine secretion. So when we think of holocrine, I want you to think sebaceous glands. The classic example of a holocrine gland is going to be the sebaceous gland of the skin that secretes sebum, or that oil-rich substance that lubricates our skin and hair. We use epithelial tissues to make membranes in our bodies as well. Most membranes are going to be made of epithelial tissues. Occasionally, we could have connective or muscular tissues make our membranes also. Some examples of connective tissue membranes include the dura mater within the central nervous system, the synovial membranes of our articulated joints, or the periosteum, which wraps around the surface, the superficial portion of the bones of our bodies. When we think of membranes in our bodies that are only made of epithelial tissues, those are more difficult to find. We have, obviously, the epidermis. That's the classic example. But one that most people don't think about is the anterior surface of the cornea and the lens of the eye are also going to be made of epithelial membranes. When we think of the skin, it's the single largest membrane of our bodies. It's also referred to, in addition to being the epithelium or the skin, sometimes it's referred to as the cutaneous membrane. Those are all going to be synonyms with each other. And we have that stratified squamous epithelium making up the epidermis, and then we have the dense, irregular connective tissues making up the dermis deep to the epidermis. Within mucous membranes, so we're still talking about epithelium right now. Our mucous membranes are going to have high concentrations of goblet cells. The shortened name for mucous membrane is mucosa, and these are typically going to line passages that open to the external environment. And these are going to be situations where we need to protect ourselves from extracellular pathogens. So every time you breathe in several hundred bacteria and mold spores, even with really clean air. It's completely unavoidable. And every moment of every day, you breathe in these mold, these bacteria, these dust particles, and you need some way to keep mushrooms from growing in your lungs. So we have mucous membranes, and the spores and pathogens get clogged up in the mucus, and then the cilia carry the mucus away, and then we dump the mucus into our stomach and digest everything with hydrochloric acid in our stomachs, which kills the vast majority of the stuff that we breathe in. So when we think of these mucous membranes, we also are going to have them in the digestive tract. And it's not that we breathe in bacteria with our digestive tract. In this situation, we eat the pathogens, or we eat the microorganisms, and we use the mucous membranes in our digestive tract to protect us from those microorganisms and to protect us from the digestive components of our digestive tract. A serous membrane, unlike a mucous membrane, is going to be a membrane, as its name implies, that makes a very watery or low viscosity excretion. When we think of a serous membrane, we can think of the serosa, those double membranes that line our body cavities. In between the parietal serosa and the visceral serosa, we have a thin, watery, slippery fluid that's a serous excretion from those serous membranes. We're also going to have a serous membrane associated with joint capsules. So when we think of, that classic joke, elbow grease, that you can't actually buy it at the store, but elbow grease, if we really wanted to be pedantic, is going to be a serous secretion from the serous membrane of our articular capsules that helps to lubricate the articular cartilage within our freely moving joints. It's a slippery, watery fluid. Something that's worth emphasizing in the margin notes, and I mentioned this with Chapter 2, is that if something feels slippery to the touch, oftentimes it's going to be alkaline or slightly basic. A lot of our serous secretions have a very slightly alkaline pH, and also are going to have some carbohydrates mixed in that are going to help to lubricate the fluid. So you can think of it as very diluted mucus. So there's a little bit of carbohydrates mixed in, a lot of water, and a little bit of alkalinity that makes it super duper slippery. So we're pretty much done talking about how to categorize epithelial tissues and epithelial glands. This in-betweeny section, tissue growth, just need to be put somewhere, so I put it right here. So we're transitioning to a slightly different topic right now, related but slightly different. Let's talk about how we can grow tissues. When we think of tissue growth, I want you to think of increasing the number of cells or increasing the size of an existing cell. When we think of hyperplasia, this is going to be growth through making new cells. Unborn kiddos, little kids, teenagers have lots of hyperplasia. When you're physically growing larger, you have lots of hyperplasia, typically, when you're in the active growth stages. And then we can have hypertrophy. We, as adults in this room, don't have a lot of hyperplasia anymore. I mean, occasionally we'll have hyperplasia, maybe if we damage our skin and we need to grow some new skin. But for the most part, we aren't growing taller, we aren't adding more brain cells, we aren't adding more muscle cells. So if you go to the gym and you bulk up, you take the existing skeletal muscle cells you have, you don't add new ones. Instead, the existing ones you have get bigger. So you increase the volume of existing cells in that example. If an adult has hyperplasia of maybe the skeletal muscle cells or osteocytes, bone cells, or hyperplasia of neurons in the brain, that's also known as cancer, out-of-control cell growth. And we shouldn't have that as an adult. Little kids are a different story. They are physically growing larger. They need to add more cells. As an adult, really the only two tissue types we're going to have hyperplasia of include adipocytes. Unfortunately, it's really easy for us as adults to add more fat cells to our bodies. It's not cancer. It's just too many calories. And then another one that we have hyperplasia of as adults is going to be the epithelial tissues of our membranes. Think of our skin, the lining of our digestive cavity, those tissue types that are exposed to constant damage. We need to constantly regrow the cells. Neoplasia is unwanted hyperplasia as an adult. And that's going to be referred to as the tumor or the neoplasm. If it's benign, it's just chilling. It's just sitting there, and it's just growing bigger all by itself. When we talk about tumors, a malignancy or a malignant tumor is one that's rapidly shedding off fragments and spreading to other parts of the body. So if you had to pick what kind of a tumor you had, definitely choose the benign tumor. They're much easier to remove from the body. If you have malignant tumors, the best course of action is to remove large swaths of tissue from your body and then give you lots of chemotherapy. So as we have our tissues in our bodies growing, these tissues change over the course of our life cycles and our lifespans. For instance, our blood, when we're brand new, freshly born, fresh out of the womb, is different from our blood as an adult. We go from one kind of red blood cell with fetal hemoglobin in it to a different kind of red blood cell with regular adult hemoglobin in it. We can change tissue types as we age. Another classic example of changing tissue types, or this difference that happens as we age, is the lining of the vaginal canal. Prepubescent young girls will have a simple columnar epithelium lining their vaginal canal, which is not designed to withstand abrasion, but post-puberty, post-pubescent young ladies are going to have a stratified squamous epithelium lining their vaginal canal, which is capable of withstanding frictional forces. So we can have differentiation over time as we age, and one tissue type can differentiate into more specialized tissue types as well. The example of the differentiation would be from an embryonic or adult stem cell becoming a specific subtype, or we could have that metaplasia with the example of one tissue type transforming completely into a separate tissue type. The example I gave for metaplasia was the simple cuboidal epithelium transforming post-puberty into the stratified squamous epithelium. Let's talk stem cells. When most people say stem cells, and when most news organizations talk about stem cells, and I'll admit this is a pet peeve of mine, they group all stem cells together. But there are different kinds of stem cells, and the implications physiologically and ethically are very different for the different kinds of stem cells. So, generally speaking, a stem cell is a cell type that can be differentiated into other cell types. So it's an unspecialized cell type. Earlier on in our development, we have more plasticity to our stem cells. We have the ability to regenerate tissues faster, and we have the ability to make more replacement tissues. We have lots of that developmental plasticity. As we age, we lose developmental plasticity. And it's not just we have it one day, we lose it the next day. This is a gradual process over the years. We lose the ability to differentiate cell types within our bodies. So let's talk about different kinds of stem cells. We have totipotent. And I think this will be our last slide for the day. I have 9:14 on my cell phone right now. So we have totipotent stem cells. A totipotent stem cell is a stem cell that could be part of a human body or some accessory organ which is not part of the human body. And most people will stop and say, what? What kind of an organ is not part of a human being? These are going to include things like the placenta or the umbilical cord. So it's still made of human tissue, but as an adult, we don't have placentas or umbilical cords hanging off of our umbilical regions. So the totipotent stem cell can be either a adult human organ or a embryonic organ. Pluripotent stem cells have the ability to become any tissue type or any cell type within an adult human being. The common name for a pluripotent stem cell is an embryonic stem cell. Back in 2008, there was another kind of cell type added. I'll get to that one tomorrow or Thursday. And then finally, the last thing we're going to focus on is multipotent stem cells. A multipotent stem cell is an adult stem cell. It's a partially but not fully differentiated stem cell. So it's kind of specialized, but it still has the ability to turn into different tissue types. So those are the three big categories of stem cells. We'll pick up on induced pluripotent on Thursday. That's all I got for you today.